Welcome and shalom to the third seminar in our Australia-Israel Scholars Seminar series. I'd like to start by acknowledging the Turrbal and Yagara people as traditional owners and their custodianship of the lands on which we meet today at the University of Queensland. I pay my respect to their ancestors and their descendants. So in the room today, we're joined by staff and students of the University of Queensland, and we have some staff online as well. And I'm thrilled to welcome Dr. Ido Goldstein from the Hebrew University of Jerusalem to our seminar today. I'm fortunate to call Ido an old friend of mine. Ido is an assistant professor at the Institute of Biochemistry, Food Science and Nutrition in the Robert H. Smith Faculty of Agriculture, Food and Environment at the Hebrew University of Jerusalem. He did a postdoc at the National Cancer Institute and the National Institutes of Health in the US in the lab of receptor biology and gene expression, and has a PhD from the Weizmann Institute of Science. Today, Ido will present on the biology of fasting and its link to our genome. So take it away, Ido.
Thanks, Joe. It's a pleasure to be there, albeit virtually. And thank you for having me. And a special thank for you supporting us in these very difficult times in Israel. It means a lot to us, the support we're getting from abroad. So thank you, everyone, for having me and for the support. And as Joe said, I'm going to be talking about the biology of fasting. I'm going to give a very brief introduction. I'm not going to, hey everyone. I'm not going to delve too much into the data. You're welcome to ask questions, of course. But I'm just going to briefly introduce what we're doing in the lab. So fasting is a part of our inherent part of our history. Actually, it's been part of every major religion. We know every major religion has some fasting incorporated into the rituals. Of course, Christianity, Islam, Judaism, Buddhism, all of those have some rituals of fasting. And actually, fasting has been part of our prehistory as well, a pretty big part of our prehistory, because it's the common conception now is that most of the times before the invention of agriculture, we were fasting most of the time, and only occasionally consumed food. We didn't have these 3 meals with snacking in between in those prehistoric times, and only sporadically consumed food. So we tend to think of fasting in prehistoric time as pretty much our basal state, the state at which we were most of the time dealing with. So our body is very well equipped to withstand fasting for a long period of time, and we developed very complex mechanisms to withstand fasting. And actually, we as humans can fast for a long time. I guess there's no microphone there, but you can guess. I'm going to give you a few seconds to guess what is the longest recorded fasting duration that you can think of. Anyone can guess? I can't hear you, sorry.
Someone said 6 months.
6 months. Higher than that.
I think there was somebody who got vitamin supplements as well, I think, and fasted for a year or something, I think.
That is correct. Actually, you're the first to ever guess that. In any of my seminars, people tend to say a few weeks, a month, 2 months. So this person did not consume any calories. So vitamins don't have calories. Of course, he drank plenty of fluid, and he fasted for 382 days, so more than a year. Obviously, this person was morbidly obese. I mean, he weighed 204 kilograms and lost 61% of his body weight. But the point is, it's not like I'm recommending this to anyone, but the point is we can survive prolonged fasting periods. We have the mechanisms to survive that. So what is the main issue with fasting? The main issue is that our cells consume energy all of the time. When we rest, when we do sports, when we sleep, most of the energy that our body consumes is completely irrelevant to the physical activity, whether we do it or not. And the only energy source that we have is food. There's no other way for us to obtain energy. So that puts us in a problem when we don't have available food, but we still need a lot of energy. So energy is stored in our body in several manners, but the most accessible way energy is stored is in fuel molecules. There's a few molecules that we term as fuels, and those molecules travel through the blood and supply our tissues and organs with the energy that they need. So obviously, there's a problem because when we don't eat, there's no energy coming from food. And that problem is relatively easily solved after a meal, because after a meal, we have plenty of energy that we're digesting, and most of it is, some of it is consumed immediately for our current needs, but most of it is stored away for times when we don't have food. So we're very good at storing energy. Most of our energy is stored in fat depots. So that's not a big issue after a meal. But when fasting becomes prolonged, the mechanism is to survive and to supply energy is to produce fuel molecules from these storages. So produce fuel molecules and distribute them via the bloodstream. And the major organ that does this is the liver. The liver is the major organ that produces fuel and supplies fuel for the rest of the body during fasting. How does the liver knows to start producing fuel? The liver does not directly sense the lack of energy. It does so indirectly via hormone signals. So there's a few hormones that are secreted from glands during fasting. Some of them, the major ones are glucagon and glucocorticoids. You may have heard of cortisol, the stress hormone. This is a hormone that is secreted during prolonged fasting. And these hormones reach the liver and signal it to start to produce fuel. And the fuel that is produced during fasting, there's two types of fuel. Glucose, which is the major sugar that our cells like to use, and ketone bodies. These are derivatives of fat oxidation, the burning of fat coming in from adipose tissue. So these are the two major fuels that the liver produces. And what we study in the lab is how we're starting with low energy balance, increase of secretion of hormones, and we're ending up with the liver producing a lot of fuel. And part of the way by which the liver does that, I'm assuming you're seeing my cursor, and if not, let me know. So these are liver cells. They are called hepatocytes. As you can see, this is one cell, this is another cell. And within the cell, like in almost every other cell, there's a nucleus, okay? Actually, hepatocytes have two nuclei. You can see that here, they look like little eyes. This is a story for a different presentation, but within, why hepatocytes have two nuclei, but within the nucleus, like in every other cells, there's our DNA. And our DNA is a very, 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 very long molecule. Actually, if we would stretch the molecule, the DNA molecule of one nucleus, just one cell in our body, we would reach, if we would stretch it, we would reach 2 meters. So we need to store a lot of DNA in a very, very small compartment, which is the nucleus. And this is done by what we call chromatin. Chromatin is the compaction of DNA with the help of proteins into very, very organized, in a very organized manner. Think about this when we just have a cable or a cord lying around on the desk. I can look now at my desk, there's a few cords just lying around for a few days. They get tangled up with one another and completely unusable. This is exactly what would happen to DNA if it would not be packaged and organized in a very strict manner. So the thing about DNA is that all cells have all of the genes in the genome, but none of the cells use all of the genes within this DNA. So most of the genes within the DNA is not used by cells because every cell type uses a different set of genes. So in every cell type, you have quite a lot of genes, thousands and thousands of genes that are not used. And these genes are compacted and packed in a very strict manner because they're not used and when we don't need them. But the regions from which genes are expressed, and when genes are expressed, they are later produced into proteins, translated into proteins, and these proteins basically give the identity of the cell. So if the liver needs to produce fuel, it needs to express genes encoding proteins, which later produce the fuel. So the gene expression is symbolized here by this brownish worm. This is the copying, what we call transcription of the genes into RNA molecule, which is then translated into a protein. So every cell type needs to express a different set of genes. What we and others have found is that upon fasting, the expression of the genes in the liver is radically changed. There's a lot of genes whose expression is induced, is increased following fasting, and other genes whose expression is very much dramatically decreased. And not only that, we have shown that thousands of regions within the genome become less compacted upon fasting, while others become compacted. So it's not only that different cell types like liver cells versus neurons or skin cells, it's not that only they have a different compaction of different regions in the genome, but within the same cell, there's dynamic changes in the genome organization, depending on the fact where we're feeding or fasting. So some regions become opened up upon fasting, while others becomes closed, and this enables gene expression. And what controls this differential expression of genes is initially those hormones that I described before. So the hormone signal that this is now fasting time. The liver, with the help of proteins, the nucleus, the DNA within the nucleus becomes opened up, accessible, and this enables the expression of genes, and eventually this is what enables the production of fuel, the production of glucose and ketone bodies from our liver cells. So I'm not going to go too much deeper into genome organization and the link between genome and fasting, but I just want to, before I finish, just want to briefly mention, I'm sure you've heard about intermittent fasting. These are dietary regimens which are becoming more and more popular. So people who voluntarily fast for either every other day, so 24 hours of fasting followed by 24 hours of eating, or people that fast for 16 hours out of those 24 hours, and then eat for the next 8 hours, which is called the 16-8 diet. And there's other variations of those intermittent fasting regimens. And there's quite a lot of studies to show that in humans, this is pretty beneficial. So reduced blood pressure in people who have hypertension, high blood pressure. In people with diabetes, this led to reduced blood sugar and reduced insulin levels, which is what we aim for in diabetes patients. People with increased cholesterol, increased triglycerides in the blood also show marked reduction in those lipids, and obviously reduced weight. This is pretty well established that intermittent fasting leads to a reduction of weight. In animal studies, where we can control the animals much more than we can control the people, we make them fast much more easily. We've seen much more striking effects. So just a few examples. Complete recovery from diabetes. Mice that were morbidly diabetic and overweight were completely recovered from diabetes. Let me remind you, currently we have no cure for diabetes. We have many treatments for diabetes, but no cure. These mice were completely cured from diabetes, type 2 diabetes. Mice with Alzheimer's symptoms showed marked improvement in Alzheimer parameters. In certain cancer models, mice were completely cured out of cancer. In others, they showed improved cancer parameters. Another disease which we have no cure for is which we found, not us, but other people in the field, complete reversal of fatty liver. Fatty liver is the most common liver disease, and intermittent fasting completely reversed that, cured it completely. And the most striking effect that we found in every animal models that we studied is increased longevity. So every animal model that we tested this on lived longer in intermittent fasting. In most cases, these animals had to fast intermittently for their entire life. So that's pretty hard to do in humans, impossible to do. But it just goes to show how beneficial fasting is. And it brings me to my first slide, back to my first slide, where I told you that fasting was pretty much our basal state in prehistoric time, and we are wired evolutionary wise to fast for a large chunk of our life. There's many other beneficial effects of fasting. I'm not going to dwell too much on it because it goes on and on. Every day there's a different paper showing the beneficial effects of fasting. By the way, I'm not an MD. I cannot recommend anyone to fast. This is completely voluntary, and there's much more to do to study why fasting is so beneficial. But it is a very promising strategy. And that's it. I'd like to, first of all, thank you for coming, for your attention. Thank you for your support for us, the Israeli academia, in these very difficult times. And of course, thank my lab members. These are my lab members. And I'm open to any questions you might have and discussion. Thank you.
Thank you so much, Ido. I think we'll have some questions from the audience. So, yeah.
I'll go first. Thank you very much. That was extremely interesting. I have a question around the glucocorticoids and cortisol in particular. The first part is there any way to induce a state like fasting through hormonal supplementation? And also, there are certain negative effects associated with higher levels of cortisol, and could they have any negative effects on fasting as well?
Yeah, so cortisol, there's many ways to mimic cortisol. Almost all of the anti-inflammatory drugs are mimicking cortisol in one way or another, because cortisol has a very strong anti-inflammatory effect. However, I would not recommend, drugging people with cortisol supplements because, as you mentioned, there's a lot of adverse effects. But this is especially due to the fact that you're chronically treating with glucocorticoids, and this chronic stress is definitely not good for you. But the anti-inflammatory effects, by the way, probably a lot of the anti-inflammatory effects of fasting are due to cortisol, because fasting has a lot of anti-inflammatory effects. This is also very well established, and probably leads to some of the health benefits of fasting. But this is acute stress. So this is a stress that begins and ends within a reasonable period of time. And this is very much different from chronic stress, where people, in our society sometimes are faced with chronic stress, even if it's not, induced by, pharmacologic glucocorticoids, but just by, psychological stress and stuff like that. This is not good for us. So chronic stress, not good. Acute stress, probably beneficial for us.
So this is something that I've previously been interested in. When I was previously studying, I tried about 6 or 9 months sort of on and off in various forms, intermittent fasting, because I was curious in terms of the experience of it. I remember looking earlier this year, or maybe it was late last year, at the topic again. I thought the research, when it came to specifically, again, this is maybe a bit more outside of the scope, but when it came to like in terms of recommendations, I thought the evidence was a little more weak. That was at least my interpretation like a while ago. I may have misunderstood or I may have misremembered. But that's kind of what I walked away from like 6 months ago when I looked at it. Um, Maybe you can help unpack or suggest sort of points, because I found it interesting. I mean, I felt different when I was trialing trialing it in different ways, whether it was like, only eating for like 4 or 6 hours, or sometimes just skipping some dinners. Particularly, I found that and having less for breakfast and then feeling better during the morning and towards lunchtime. But yeah, it seemed like that the evidence 6 months ago was sort of not so strong. So I'm just curious how that pairs now with your suggested about the effects on animal models, and yeah, help unpack that a little bit.
Yeah, so there is a lot of discussion about whether it's beneficial or not. There's a lot of issues with making a clear statement. I mean, probably the consensus is that if you're talking about weight reduction, yeah, it's beneficial. All of the other effects are a bit more controversial, but I would say, because as you mentioned, it's every study did the fasting a bit differently. Probably the best suggestion I have is something along the lines you said, which is skipping dinner rather than skipping breakfast. And pretty much if it's dark outside, don't eat. If it's light outside, you can eat. Pretty much that would be my rule of thumb. It depends what you check. So some studies check, heart disease. Some studies check diabetes-related effects. Some people really suffer from fasting, so they can't stand it, and obviously that might lead to bad effects. Our lab does not do any clinical studies, right? So everything I'm telling you is from the literature, from me reading the literature, and I think that while some studies questioned it, none of them questioned it. So they said, well, it's not better than just dieting, just reducing your calories. But nobody that I know of said it's really, really bad for you, right? So they just said, okay, you don't need to fast, just eat less. It will give you the same effect. That's very arguable. When I talk to people doing clinical studies, some of them are pretty angry about the other people saying that, because they said, well, they didn't do it correctly. If you do it correctly, the clinical trial, you will see the benefits.
That actually seems to be how I remember it now. That seems right, because I think it was sort of saying, I wasn't reading primary research, it was sort of secondary publications around science communicating with the public. And I think that was sort of like the consensus was like, it may be beneficial, but it's not quite there yet. But I can see how, for example, with diabetes, it's potentially a thing, or if you've got hypertension or hyperlipidemia or any sorts of other health issues. Yeah, I'm wondering, could you touch a little bit more on sort of the specific type of research that you do? Is it more in the genomic space, I think? And if so, like, what are the things that really excite you at the moment in your lab or in your field? Because obviously genomics is like this big beast that sort of people are working on from different angles, and there's more tools coming out, and there's all this AI things coming out. Like, what are you excited about, and what are you working on?
Yeah. So yeah, basically I come from a molecular biology background. I'm not coming from a metabolism or more clinical background. And what we study in the lab is basically how genes are expressed. How do our cells decide which gene to express now and which gene to shut down? We love the fasting model. This is our pretty much our only model now in the lab, but it's a model for us to study gene expression and gene regulation. And we study specific proteins, which I did not talk about. They're called transcription factors. Those are the factors that bind DNA and determine which genes turn on and off. This is what we are really excited about. And one more avenue is, which touches about your previous question. I think that intermittent fasting is not going to be, it's not going to work for everyone, and people are going to dwell about whether it's good for you or not good for you for ages now. So, and I basically, I don't think we can tell someone, okay, fast every other day for the rest of your life and you'll be healthy. Almost nobody's going to do that. So what we are aiming at, and this is for the long run, we're not anywhere near it in the lab, is trying to understand the biology of fasting better, the physiology of fasting. So not the pathologies around it, like obesity and diabetes. The physiology of fasting, we want to understand it better so we can mimic at least part of it in a druggable pharmacological way. So if I would tell someone, for example, fast for only 12 hours a day, that's pretty easy for most people to do because we sleep at least half of that time, right? Fast for 12 hours and we're going to give you another supplement or a drug that is going to make it as if you're doing, for example, alternate day fasting. So as if you're fasting for 24 hours. If we can tune in the fasting response and make it stronger, make the body think we're fasting for longer periods of time with fasting only overnight, that I think is the main approach that we're pursuing. And we're pursuing that strictly by looking at the very basic biology of fasting. How does it work, basically?
Thank you for taking time out of your day to come talk to us. I'm sorry if my question is a bit basic. I don't have a science background. I'm just came because I was interested. But I was interested in hearing about the effects of after these people who have undergone quite long periods of fasting, how that might have influenced, cravings and their long-term ability with digestion and their microbiome. If there's any research on that. Sorry if this question sounds a bit basic.
Oh, yeah, I get those questions a lot. So some people cannot stand it. I mean, some people are really, have cravings and hunger. For most people, that goes away after like a week or two. They get used to it, basically. But some people don't. Some people just suffer. And I can tell you, I'm not very good at fasting at all. I mean, I'm not the best example. In terms of the microbiome, it definitely affects the microbiome. On the other hand, everything changes the microbiome, so we don't really know if the changes in the microbiome are really linked to the beneficial effects of fasting. I'm not saying that they don't. It is very reasonable to assume that they have some effects. But right now, we just know there's a link. The microbiome, the fasting affects the microbiome, but we don't know if more than that, basically.
Thank you so much. That was such a fascinating talk. A big round of applause from us all here. Thank you again for joining us, and thanks for everyone coming. We're holding these seminars twice a week, so please get in touch with myself or Yoni if you have an idea for a future speaker. And thanks again, Ido, for taking time out of your day. And shalom, everyone.
My pleasure.
Bye.
Thank you.
Bye.
Bye-bye.
Bye.